I want to say just a couple of things about retroviruses. Retroviruses are defined as viruses that use RNA as their genetic material, that are capable of inserting a DNA copy of their genetic material into the host cell's DNA. Now, the best studied example of a retrovirus is the human immunodeficiency virus, HIV, the virus that causes AIDS. The reason I want to talk about it is because this virus uses a very peculiar enzyme called reverse transcriptase, or RT. I mentioned RT in the Baltimore system. One of the ways that the Baltimore system classifies viruses is by whether or not they contain reverse transcriptase. So, what is reverse transcriptase and what makes it so unusual? We have to recall the central dogma here. Remember the first statement in the dogma. It says DNA is used to make RNA through a process called transcription. Reverse transcriptase is an enzyme that can do transcription in reverse, essentially. Reverse transcriptase can make DNA from RNA, which is the reverse of how it normally happens. So this virus uses RNA as its genetic material, but when it inserts that RNA into the host cell, it also sends along this enzyme. And inside the host cell, the enzyme takes the RNA and makes a DNA molecule out of it. That DNA is then inserted into the DNA of the host cell. The host cell, again, can't tell viral DNA from its own DNA. So the host cell doesn't do anything when this viral DNA gets placed into its own DNA. The host cell just treats it all like its own DNA. It replicates its DNA every time it divides, and it uses its DNA to make proteins. So it's going to be making viral proteins as it makes its own proteins. Now, reverse transcriptase is packaged into the virions, into those extracellular viral particles, and it's pre-made, in other words. So this is an unusual thing. We don't see this in most viruses. Inside the capsid is not just genetic material, in other words. Genetic material is in there, and this enzyme is in there, this reverse transcriptase. One of the things that makes human immunodeficiency virus so terrible is the fact that it's a retrovirus. When it inserts its DNA into the host cell DNA, it just sits there. It just sits there for sometimes years. So the person doesn't know that they are infected with HIV until that HIV virus sort of reactivates inside the host cell and does damage. So let's talk a little bit about what happens inside the host cell. We've talked about how the virus gets into the host cell by binding to a receptor. And we've talked about how the host cell makes copies of the genetic material and builds the viral proteins so that new viral particles can be made and then leave the host cell and go on and infect other cells. We've also talked about how some viruses, including retroviruses, can just stick themselves into the host DNA and just sort of sit there for a while. So let's think about these life cycles in a little more detail. Bacteriophages, remember those are the kinds of viruses that can infect bacteria. They can have two distinct life cycles, two types of growth cycles, if you will. What we call a lytic cycle and a lysogenic cycle. So those are terms we should be familiar with. When you call a viral life cycle a lytic cycle, what you're saying is that at the end of the cycle, the viral particles are going to burst out of the host cell. They are going to lyse or break the host cell and, of course, kill it as they exit. That's called a lytic form of viral cycle. A lysogenic viral cycle is different. That's the term we use when we're thinking about a virus that can insert its genetic material into the host's DNA, sometimes for long, long periods of time. The thing about the lysogenic type of cycle, again, with HIV as a good example, is that eventually, that genetic material, that viral genetic material, will come back out of the host's DNA and it will go into the lytic cycle and eventually leave the host cell. So, those are the two basic life cycles that we see in bacteriophages. And remember, when we talk about lysogenic viruses, the retroviruses are only one of the lysogenic viruses. So, when it comes to the bacteriophages, the end result of either the lytic phase or the lysogenic to lytic growth cycle, life cycle, in both cases, the viral particles exit the cell by killing it, by breaking it open or lysing it and killing it. Eukaryotic viruses are a little bit different. Yes, there are lytic and lysogenic viruses that infect eukaryotes, yep. But there are some eukaryotic viruses that don't kill the host cell when they leave. Some eukaryotic viruses can bud out of the host cell. Some of them can use a cellular process called exocytosis to get out of the host cell. Both budding and exocytosis don't kill the host cell. The viral particles get out to go on and infect other cells, but they don't kill the host. Here's a depiction of those different ways of getting out of the host cell. Over on the left, this represents that process called lysis. That's a very generic word. It just means rupture or break. So we can see here's a viral particle, and it is exiting this sort of an orangey peach colored host cell with its membrane shown in gray, and it's going out into the environment. On the way, it is breaking the membrane, so it's killing that host cell. All of the bacteriophages, all of the viruses that infect bacteria, will kill the host when they eventually leave. Some of the eukaryotic viruses also do this, also exit through lysis. But some eukaryotic viruses get out of the host cell without killing it. They either use exocytosis, which is a host cell process, or they simply bud out. So by budding, we mean they push out through the membrane. They actually take a piece of the membrane with them when they go. And as they bud off and exit, the other pieces of the host cell membrane just repair. They just come together and seal themselves. This budding process is how a viral particle gets an envelope. And remember, we said we see enveloped viruses primarily in animals, eukaryotic animals. Now, viruses that exit through exocytosis don't get an envelope. And that's because they are hijacking a natural process in the cell. All exocytosis is the process by which cells excrete things or secrete things. They make materials, and they're showing us here the endoplasmic reticulum and the Golgi apparatus and a transport vesicle coming and merging with the membrane and releasing its contents. So the viruses just hijack that natural process as a way to get out of the cell. I've got a lot of pictures on this slide that you may recognize. Up in the upper left-hand corner, we see this young man who happens to have a good case of the chickenpox. If you had chickenpox as a child, you know that the chickenpox virus happens to belong to the herpes family of viruses, and it causes this pox disease. You get these blisters all over your skin, all over your body, and they eventually rupture and scab over. That's what you're looking at here in this image. Very common childhood disease. It is typically not a particularly pathogenic virus. It's uncomfortable, certainly for the child or the person who gets it. It's uncomfortable. It can be a little disfiguring because you can end up with pox marks on your skin. But it's typically not a fatal virus or anything. Now, herpes viruses are a form of eukaryotic virus that belongs in this lysogenic category. Herpes viruses, when they get into human host cells, they integrate their genetic material into our DNA. So while you get sick with chickenpox virus when you get it, yes, the next thing that happens is that virus leaves itself, imprints itself, inserts itself into your DNA, and it stays there. It doesn't bother you. It doesn't harm you. It happens to go into and insert itself into cells without you really realizing that it's happening. But even after you recover from chickenpox, you are carrying the chickenpox genetic material in your DNA, in some of your cells. And for many of us, it never bothers us again. It never goes back into the lytic cycle, in other words, and never exits our cells. But for some of us, the chickenpox virus does reemerge. And that's what you're looking at on the right-hand side. This is a disease called shingles. And if you've ever known anybody to have shingles, or you've ever had it yourself, it's not like chickenpox. It is a painful rash. It tends to not only have the sort of clear serum associated with it that it comes out of the blisters when you have chickenpox, but it actually bleeds. So people who get shingles will talk about having this wet rash that is sticky, sometimes bloody, and very painful. Horrible, horrible disease to have to deal with, shingles. What's happening in shingles is, for some reason, and that reason relates to stress, for some reason in the host's life, the virus exits the host cell DNA. So the viral genetic material comes back out of the host cell DNA and enters back into the lytic life cycle. So it's lysogenic for a while, it sits in the DNA for a while, and then it reverts to the lytic life cycle, and it exits the host cells. Not everybody gets shingles because not everybody goes through the physiological stress that causes the virus to come back out. What kind of stress causes shingles? Well, there are a couple that are well documented. One of them is the stress. Again, we're talking about physiological stress here. We're talking about biological stress. The stress of aging can do it. In some people, as they age, their immune system starts to really slow down. Their immune system starts to work less well. And that kind of stress can push that virus back into the lytic growth stage and cause shingles. For some people, it is another disease process that's going on. It's the stress of chronic disease that will cause shingles to occur. And again, for many people get chickenpox as a child and then live the rest of their life and never get shingles. And that's because they didn't go through enough of a stress process anywhere that caused the virus to come back out as shingles. So let's take a look at some artist depictions of lytic versus lysogenic, because those are the two growth stages I want you to be familiar with, or the life cycles I want you to be familiar with. Remember, if you use bacteriophages as your model, bacteriophages can be either lytic or lysogenic. But lysogenic bacteriophages can revert back to lytic when they experience certain stressful events, or when the host experiences certain stressful events. Remember, eukaryotic organisms, the viruses that infect them, can also be lytic or lysogenic. It's just that eukaryotic viruses can exit cells through different mechanisms. All right, so here's an artist's depiction of the lytic life cycle. And again, the artist is using a bacteriophage as their model. So this is a bacterial host cell, and what we're looking at in pink is that circular bacterial chromosome, their piece of DNA. The bacteriophage is going to infect this cell. It's going to bind to a receptor on the surface of a cell, and this particular virus injects its genetic material, and that's what they're showing us here. It's going to inject its genetic material in green into the host cell. Now, that injection process is kind of fascinating, because what's going to happen is that viral DNA is going to come in. It's actually going to cause the bacterial chromosome to sort of break apart into pieces. We don't need to go into too many of the details of that. But the host cell is going to treat the bacteriophage genetic material just like its own genetic material. It's going to copy it. It's going to make proteins from it. It's going to build new viral particles. You're going to get new bacteriophages, in other words, built inside the host cell. And when the time is right, those bacteriophages are going to lyse the host cell and exit. The host cell then dies because of that lysis, because the membrane gets broken. Now let's look at the lysogenic life cycle. Here's another bacteriophage, and it's injecting its genetic material into the host cell. This time, that genetic material is getting placed into the host cell DNA. You can see the green piece now inserted into the pink piece. So this is a very different process. And once it's inserted in there, once it's incorporated in there, it can stay there for a long time. The bacterial cells can divide. And again, in order to divide, in order to go through that process, they have to copy their DNA. And they copy the viral bit right along with their own DNA and pass it on down to the next generation of bacterial cells. And this can happen again for years. It can just keep happening. It just stays where it's sitting in the host DNA. Now, something sometimes can happen. It doesn't always happen, but something can happen that stresses out the host cell. Now you say, well, what kind of thing stresses a bacterium? Remember, things like a lack of water, desiccation, that's called. Desiccation will stress out the host. A lack of nutrients will stress the host. Too much waste material in its environment will stress that host. And when that happens, the viral DNA can come back out of the host cell DNA. And when it does that, we're going to go right into the lytic cycle. That exiting is going to cause the bacterial DNA to break into pieces. It's going to get used to build proteins along with the viral DNA. We're going to get new viral particles made. They're going to rupture and exit the cell. So sometimes during the lysogenic cycle, we stay in the lysogenic cycle. And sometimes the lysogenic cycle reverts back to the lytic cycle. So when it comes to the prokaryotic viruses, and remember by prokaryotic we mean bacterial and archaeal, the life cycles that we're seeing are the lytic and lysogenic type. And regardless, we're talking about those viral particles exiting their host cell through lysis. The eukaryotic viruses are different. Some of them exit using lysis, some of them don't. So I talked about one of the herpes viruses, the one that causes chickenpox specifically, being a lysogenic type of virus that sometimes reverts to the lytic life cycle, exits the host cell through lysis. But remember, there are other eukaryotic viruses that use either budding or exocytosis as a means to get out of the host cell without killing it. And for these types of viruses, for viruses that are capable of exiting without killing the host cell, it includes a whole range of viruses such as rabies and the virus that causes measles and mumps, the human immunodeficiency virus, the smallpox virus, the SARS viruses, including SARS-CoV-2, some of the herpes viruses, and the hepatitis B virus. So when it comes to the eukaryotic viruses, there's a real diversity in how those viruses exit the host cell. Lysogenic viruses they're fascinating because they are capable of establishing this long-term relationship with the host cell. They don't come into the host cell and get rapidly replicated. Instead, they go into the host cell and they just essentially become part of it. If you think about it, when viral DNA gets inserted into the host cell's DNA, they're essentially putting their genes in and among the host cell's genes, and they leave them there sometimes for long periods of time. So that means that not only is the viral DNA replicated or copied every time the host cell divides, but the viral DNA is being expressed. It's being used to make proteins. That means that those host cells are getting viral proteins made without going through making new viral particles. Viral genes can get passed down through generations of the host cell. It's really quite fascinating. And unless that host cell undergoes some kind of stressful event that causes the viral DNA to revert back to the lytic life cycle, Sometimes it stays there forever. When you look at the human genome, when you look at the human DNA molecule, and remember there are somewhere in the neighborhood of 30,000 genes, right, in the human DNA molecule, somewhere in the area of 8 to 9% of that DNA molecule is viral in origin. In other words, our human DNA has viral DNA in it that was inserted who knows how long ago. Viral DNA that humans have picked up over the millions of years that we've been on the planet. Viral DNA that has been inserted into our human genome that has never left. 8 to 9% of human DNA is actually traced back to viral origin. It's kind of fascinating.